Okay, hello again, everybody. Thank you for your patience. I think we're ready to begin now. So thank you all for logging on to Grand Rounds presented by the Department of Psychiatry and Behavioral Sciences. And I wanted to let you know that today's presentation is sponsored by the Garvey Institute for Brain Health Solutions. So thank you to the Garvey family for making this presentation possible. My name is Jed Thompson. I am your host of Grand Rounds. I want to do a few quick notes before we begin. Actually, let's go to the next slide here. As you can see, we have our.
You have to use the mouse for first.
Oh, the mouse. We have a great, exciting guest with us here today, and I mean with us quite literally. As you can see, we're coming to you from the BB Tower on UW of Washington campus, and we are going to have a little bit of time for Q&A at the end. So if you can think of any questions, go ahead and type those into the Q&A. We'll queue those up, and then I'll read them at the end of the presentation. If you would like to provide any feedback for us at the end, there will be a survey link that will show up after the webinar closes. And I wanted to let you know our next presentation is October 3rd, next Friday, with Alicia Thompson and Shannon Simmons. So please join us again for that. So with that, I'm going to hand it over to the chair of our department. To my right here, Dr. Jürgen Unitzer, and he's going to introduce us to today's speaker. So Jürgen.
All right.
Take it away.
Thank you, Jed. Good afternoon. Welcome to Grand Rounds in Psychiatry and Behavioral Sciences. And today I have the pleasure of introducing our speaker, Dr. Jeff Daskalakis. Jeff is going to talk with us about harmonizing effectiveness and physiology, exploration of magnetic brain stimulation in depression. Before I turn it over to Jeff for his presentation, I'm going to say a few things about our speaker today. Dr. Daskalakis is the chair of the Department of Psychiatry at UC San Diego. He's also the inaugural holder of the Igor and Joanne Grant Endowed Chair in Psychiatry there. He moved there a couple years ago from the Center for Addiction and Mental Health at the University of Toronto, where he was the inaugural holder of the Temerty Chair in Therapeutic Brain Intervention. Jeff's research is focused on understanding the neurophysiology underlying psychiatric disorders and how novel interventions like magnetic stimulation exert their therapeutic effects. He's had almost $100 million in grant and contract funding from federal and other sources, and he's got more than 550 peer-reviewed publications. He's also an editorial board member and the editor of several journals, including Biological Psychiatry, and he's a member of the Scientific Council of the Brain and Behavior Research Foundation. That's the old NARSAD. Now, as you all know, we have a new center for neuromodulation at UW Medical Center Northwest, and it's got some state-of-the-art equipment for TMS, so it's really great for us today to hear from one of the world's leading experts in magnetic brain stimulation about this relatively new treatment modality and what potential it may have for us as we try to help our patients with treatment-resistant depression and other serious mental health disorders. So with that, please join me in extending a very warm welcome to Dr. Daskalakis.
Thank you. Thank you for the kind introduction. It's a pleasure to be here amongst you all. I've never been to Seattle. This is my first time, so I was looking forward to this meeting and looking forward to getting a profile of the department and meeting you all. We'll start my disclosures. I happen to be a recipient of federal funding both from Canada and the US. And I also do serve on the scientific advisory board for Brainsway, but I have received support from MagVenture for our TMS and magnetic seizure therapy trials, and have also participated, not in the US, but in Canada. We participated in industry-sponsored clinical trials using the Brainsway device that you're all familiar with. Okay, we'll start with depression. This is a famous slide from the STAR-D clinical trial. The trial cost $50 million. And what STAR-D really, I think, identified was this concept of treatment-resistant depression. That is, after 2 trials of antidepressant medications, the ability to achieve remission drops off significantly. In fact, it drops off so profoundly, as you can see there in this slide, that it's probably no better than placebo. And that's a provocative statement, but I'll actually show you some data that suggests that that's absolutely true. And so folks not only do they after 2 failed trials, not only do they struggle to reach remission, but even if they did reach remission, the likelihood of staying in remission over a prolonged period drops off significantly. In other words, even if you're taking your medications after you fail a couple of trials of antidepressant medications, the likelihood of your depressive symptoms returning is quite high. The slide at the bottom, or the piece of the slide at the bottom, is actually a sticky note. It's a sticky note that was dropped on my desk, and I couldn't help myself. I had to take a photo of it and include it, because what it, I think, exemplifies is the number of medications that get added on and build in patients with treatment-resistant depression. That is, if you don't have proper tools, what you end up doing is putting on medication after medication after medication, and leaving people with a lot of side effects. And you can see there, one's on pregabalin, I think it's 450. Another one is a lorazepam, 6 milligrams a day with a stimulant, and trazodone, and bupropion, and acetal. I mean, it goes on. And I was baffled when I saw this medication cocktail. So this is what we often see when we're dealing with treatment-resistant depression. I'm sure sometimes you see this in your own clinical practice, particularly when you get referrals from the community. So this is the slide. Brett Jones was, I should say, now he's faculty at the University of Toronto. He was my research resident. I happen to have the good fortune of working with some really exceptional trainees along the way. It's actually been, I think, one of the best parts of my career, and I have highlighted them at the top of the screen. So you'll see lots of slides with photos on them of the people that I had the privilege of working with. But in 2015, Brett and I thought, well, why don't we really try to identify what the placebo response rate is in treatment-resistant depression that had not been considered at that point in time. And the reason to consider it is we have open label trials. We were conducting an open label trial of magnetic seizure therapy at the time. I'll show you that data in a second. And we really needed to know, rather than going through the arduous process of randomized people to active medications versus placebo medications, not that you can do away with randomized control trials, but at least if you're dealing with large-scale placebo-controlled analyses, you can ascertain what the placebo rate is to give you some kind of estimate as to what you can expect versus active treatment, and where the distinction lies. So what we found, really in a nutshell here, I won't go through all the data, but what we found is the placebo rate in treatment-resistant depression is about 13%, which is ironic because it's exactly what the remission rate of depression was after 2 failed trials of medication. They literally mapped onto one another, which is why I made the slightly provocative statement that after 2 failed trials of antidepressant medication, you're not achieving remission at rates that are better than placebo. Helena Kim, also a real talented. Helena was the type of trainee. She was a first-year resident when I worked with her. You come up with an idea, and she was the type of trainee that would have a paper to you in a month, fully written, requiring very few edits. And what she looked at was the efficacy of antidepressants, not just in depression, but the efficacy of antidepressants in severe comorbid psychiatric and medical condition. This is obviously quite important because we see patients in our clinics with comorbid psychiatric disorders, comorbid personality disorders, et cetera, but also germane in the context of CL psychiatry. And I actually practiced as a CL psychiatrist up until about 3 years ago. Not that I was trained in CL psychiatry, but it happened to be the first residency that I did. It's the first part of my residency when I was back in Toronto, and saw lots of patients being put on antidepressant medications in the context of CL, both for anxiety and depression. And what we found was, again, a significant drop-off in the efficacy of antidepressant medications when you're dealing with comorbid severe psychiatric and medical conditions. So in other words, the medications, not only do they not work after 2 failed trials, but they don't work in patients who have severe comorbidities. Corey Weissman was a resident in Toronto, worked with me for 5 years. We were very lucky because, for 2 reasons. Corey was born and raised in Toronto, but Corey had US citizenship through his father, which and he was also very eager to move to the US. So I brought him with me, and now he's the director of our neuromodulation program at UCSD. And Corey's been an exceptional person to work with. Corey reanalyzed the STAR-D data. We asked the NIMH through the data archive process to obtain the entirety of the STAR-D data set. And it took us a year to wade through it. It was basically a data dump into an Excel spreadsheet. And one of our postdocs, who's the second author on this paper, spent a year trying to reorganize the data to make it interpretable. And the specific question we were interested in was not obviously to replicate or to redo rather what they did in STAR-D, but to look specifically at suicidality. So in other words, we know that what the STAR-D data outcomes are in relation to depression, but what happens if you have comorbid suicidality, conflating the clinical picture? And what we found was very similar to what I just presented in the other groups, that if you have depression with suicidality, you're less likely to respond to antidepressant medications. And in fact, if you have suicidality at baseline, medications do not work very well after the first failed trial. It's really the first failed trial where you get a significant effect specifically on suicidality, but after those failed trials, the drop off considerable. In other words, your depressive symptoms don't respond if you have comorbid suicidality, don't respond that well. And if you have comorbid suicidality, antidepressants have, after the first failed trial, very little effect on suicidal symptoms. Okay, well, you all know what that is. It's electroconvulsive therapy. I've been practicing electroconvulsive therapy. I was speaking to some folks yesterday, and they mentioned in some institutions the polarization between transcranial magnetic stimulation, ECT, and that was certainly the case in Toronto. We had an ECT group, and I had just started this nascent TMS program, but the person who was running the ECT service was kind enough to bring me into their ECT service, and I started to do ECT, and that's actually brought us together over the long run. I ended up, as he stepped away, I ended up taking over the ECT service and allowed us to do really terrific things. ECT is very effective for treating depression. These are estimates of the efficacy both with bilateral and bifrontal TMS, ECT and right unilateral ECT, efficacy over 50% in terms of remission. But ECT is not for the faint of heart. It's often not accepted. Only about 1% of people with treatment-resistant depression, not depression, but treatment-resistant depression, will go on to get ECT. It's a very small and select group of people, so it doesn't generalize very well, unfortunately. Hopefully, we'll be able to close that gap. But transcranial magnetic stimulation, as you all know, was introduced for the express purpose of being able to deliver magnetic stimulation to the cortex and achieve remission in patients with treatment-resistant depression and not having to expose them to ECT. I won't belabor the entirety of the introduction, but these are just data, meta-analysis data by Marcelo Berlim, a faculty member at the university, at McGill University. We worked together to do one of the first meta-analyses of TMS and depression that showed it had a remission rate of around 25% and a response rate of just over 30%, which is a little muted compared to the data in ECT. But these were data that related to high frequency 10 hertz, the old form of ECT, and not data. This is a meta-analysis from 2014, did not include any statistics on the new forms of TMS. And the new forms of TMS are theta burst stimulation that I'll get to in a second. This is just data from one of our trials. And in this particular trial, what we did was we used the old forms of TMS, that is 10 hertz to the left side and 1 hertz to the right side, as a bilateral form of treatment. But what we were really interested in is what happens if you extend the course of treatment, and what happens if you start to pinpoint. So in TMS, the target is of significant relevance, and now it's become even more so when we're looking at biological targets of treatment response. But this is just an early study from our group. One of the first studies that we had done just to dip our toes into the field of treatment-resistant depression. Corey analyzed that data plus another data set from 2012 Dan Bloomberger was the first author. Dan was my fellow, and now Dan is the Temerity chair. He replaced me when I left in Toronto, and Dan is one of the not just local leaders, not just national leaders, but Dan is really an international name right now in the area of neuromodulation. I had the privilege of helping train him and then working with him. And we worked with Corey to look again, not in depression, but in suicidality. And we actually found that the remission rates, as they related to suicidality, were twice as high as they were in depression. So suicidality in and of itself is a phenotype that can respond to TMS, unlike with antidepressant medications. So when you're seeing patients with resistant depression who have suicidality, please keep in mind that TMS works very effectively, specifically for suicidality, and there's lots of physiological reasons why that
ECT does as well.
as ECT does as well. You're absolutely right. In fact, I'm glad you mentioned that, because early in my career when I was doing ECT, I had a patient with bipolar disorder who had severe depression and suicidality, and we were giving him ECT. And at that time, I wasn't paying attention. I was paying attention clinically to his suicidality, but I wasn't paying attention as a singular construct. I was more interested in treating his depression, and we treated him and treated him. He didn't want to get ECT. He kept stopping it, and his suicidality would return. And at that time, aripiprazole was starting to emerge as a treatment for resistant depression, and so he really wanted to stop ECT. I thought, okay, well, aripiprazole is a reasonable therapeutic option. Let's put you on aripiprazole. 3 weeks later, he killed himself. All this to say that ECT at the time, the lesson I learned from that was that ECT was probably keeping him alive, and it's certainly the approach that I'm taking right now. But you know, it came at a cost, unfortunately, to him. Theta burst stimulation is a new form, as I mentioned, of stimulating the cortex. It involves a triplet pulse delivered at 50 hertz. So 3 pulses delivered at 50 hertz. And those triplet pulses happens really quick. You can barely hear it on the machine. It's a triplet pulse, and it is delivered for 2 seconds, and then there's a pause, and then it is delivered for 2 seconds again, and then there's a pause. And theta burst stimulation was developed originally in the mid-2000s, not as a treatment tool, but as a neurophysiological tool. So this paper shows the induction of long-term potentiation, neuroplasticity with theta burst stimulation. The bottom line, you don't have to pay attention to for now, but the top line just is a physiological, and actually I did a lot of my PhD work using these neurophysiological methods, looking at the motor evoked potential, stimulating the motor cortex, and with a train of intermittent theta burst stimulation, which is the 2 seconds on, 8 seconds off, 2 seconds on, 8 seconds off. When you do that for about 3 minutes, you can induce neuroplasticity in the motor cortex. And so there were some interesting studies that were followed up on this, but the most important of these was this paper in 2014 by Lee, which showed that bilateral theta burst stimulation, which takes only 9 minutes to deliver, produced significant effects. I think the remission rates with TMS using the bilateral theta burst stimulation approach were over 50%, albeit in a smaller study, only had about 80 patients in it, but it did show profound therapeutic effects. And the second arm, which is the 42.3% arm, is intermittent theta burst stimulation only. So these were delivered at 1800 pulses and delivered the exact same way that they had used to produce neuroplasticity in the cortex in the original paper. So it suggests that stimulating the dorsolateral prefrontal cortex induces neuroplasticity, and that neuroplasticity is key to producing therapeutic effects in depression. So that was the first important paper. The second, which was perhaps one of the most critical, is the paper by Dan and Jonathan. Dan was the first author, Jonathan's the last author. Both of them had trained closely with me. This was a multi-site trial, but we called it a multi-site trial. It was literally 2 sites at the University of Toronto that were a few miles apart from one another, but we had created this machine called the Temerity Center, thanks to the very generous donation of the Temerity family, who then went on and gave $250 million to the University of Toronto and have the medical school named after them. They're a wonderful family. I know them quite well. But they gave us the money. So they gave us the resources and the support to be able to run this pivotal trial. And this trial led to the FDA approval of theta burst stimulation for depression. And what it was a non-inferiority clinical trial comparing theta burst stimulation to 10 hertz, the standard FDA approved treatment. And because it was a non-inferiority clinical trial, we were trying to demonstrate that the two arms were identical to one another, which, as you can see plainly there, there was absolute concordance between those two treatment outcomes. So the non-inferiority statistics worked out, and what we were able to do is report this to the FDA, which then approved ITBS. Ironically, ITBS still hasn't been approved in Canada, the place where we have conducted that trial, but it's approved and is the reason why a lot of centers in the US now use, including our own, now use theta burst stimulation to treat depression. Now, the importance here is that not only is it engineered to induce neuroplasticity, but it takes a fraction of the time. Standard TMS used to take 30 minutes. Now they've shortened it down to about 20 minutes, but theta burst stimulation can work as little as 3 minutes. So it actually allows you to treat a higher number of patients over a short period. So these are just the clinical outcomes, response rates of around 50%, remission rates at around a third. And this has stood the test of time. Another trial, again, the first trial was called the 3D trial. This was the 4D trial. This was specifically in geriatric depression. Again, it met non-inferiority criteria. And you can see there the remission and response rates between the two groups. Theta burst stimulation was, for all intents and purposes, non-inferior, in fact, a little bit better than the standard TMS. But this was delivered as a bilateral treatment. So remember, I told you about the bilateral treatment that LEAD did. So this was a bilateral treatment for patients with late life depression that was published a few years ago in JAMA Psychiatry. So not only is it an effective treatment for adult depression, but also very favorable treatment response in late life depression. There's been some emerging work using the theta burst stimulation. So the benefit of theta burst stimulation is take minutes to deliver. And so Nolan Williams, this very clever psychiatrist who is at Stanford, who I'm sure you all know, he created this protocol called the SAME protocol. So the SAME protocol did some really clever things. Number one, Nolan fundamentally understood that you could deliver many treatments over the course of a day. And so he compressed all the treatments into about a week. And so in the original protocol where the FDA approved to treat depression involved 30,000 stimuli over the course of many weeks. So Nolan took every single one of those stimuli, equated it to theta burst stimulation, and then delivered that theta burst stimulation over the course of a week. And so basically what it involves is theta burst stimulation every 50 minutes. And the reason he picked 50 minutes is that there was this compelling paper, albeit in tissue models, that show that if you space theta burst stimulation every 60 minutes, you get maximal neuroplasticity. It turns out that you don't need 60 minutes probably to do that, but we haven't tested that clinically in depression, whereas Nolan did. So he spaced them about 50 to 60 minutes apart. And what the other important component to Nolan's therapeutic effects was he wanted to target this connectivity between the dorsolateral prefrontal cortex and the subgenual cingulate. And there was some suggestion at the time that the subgenual cingulate was a key hotspot in terms of biological signature in depression. And so Nolan reverse engineered a target in the cortex to be able to suppress hyperactivity in subgenual cingulate. So the two components of this trial were theta burst stimulation over multiple time points in a day, over multiple days in a week, but only a week's worth of treatment, and then targeting the specific neurocircuitry that is actually proprietary. So we don't know where he specifically targeted, but he has the code and has launched a company, I think, called Magnus as a result of that treatment. And now Magnus is I think they are using, you can purchase rights to use their product to treat depression. It achieved FDA breakthrough status in the fall of 2022, which basically means that it's being utilized at the FDA and CMS. I think will agree to fund some of it, but it has to come under very, very tight constraints. The SAINT trial was initially an open label trial, but then there were a couple or one at least, randomized trial, albeit in about 30 subjects, showing considerable differentiation between placebo. So the active treatment resulted in a significant reduction in depressive symptoms. I think the response rate was over 60%. So a real favorable profile, albeit in a small number of subjects, and I'm not sure what the status is for them locally, if they're planning to expand it, clinical trials. We have collaborated with them a little bit on this work. So I spoke about the subgenual cingulate. It is certainly a hotspot for depression. That doesn't come from us. It actually began from work way back in the 1990s by Wayne Drevitz, Helen Mayberg, who identified both in PET studies at the time, pivotal PET studies showing hyperactivity in the subgenual cingulate from FDG PET. And as a result, what they did, what Helen did specifically, is they then capitalized on this target and inserted electrodes into the subgenual cingulate with the hopes of being able to treat depression. That was the famous Brodin trial, which I'm sure, I don't know if you folks participated in it, but unfortunately, it had a futility analysis. It did not pass the futility analysis, and the trial was stopped. That doesn't imply that the subgenual cingulate is not a treatment hotspot for depression. It just implies that if you put a probe into the subgenual cingulate, the desired outcome may not necessarily be achieved. But it's still a considerable treatment target of interest in the treatment of depression. So how do you ascertain activity in the subgenual cingulate? Well, clearly you can do it through PET, which is what I show you here. You can also do it through functional MRI, but lastly, you can do it physiologically. And so way back in 2000, I was working on my PhD, and I was stimulating the cortex to try to ascertain neurophysiological signals from the motor cortex specifically. So I was doing these, what I thought were really cool studies, looking at EMG over the hand muscles, stimulating the motor cortex. One of my PhD supervisors was a neurophysiologist, so I learned neurophysiology from him. Sometimes it's fortuitous how things work out. Robert Chen, who was already at the time a leader in neurophysiology, moved from NINDS up to Toronto, took a job at the Toronto Western Hospital, which was a couple of miles from where I was located. I was introduced to him and thought this is the greatest thing since sliced bread, at least from my neck of the woods. And so I embarked with him and Satish Kapoor on a PhD looking at neurophysiology, specifically in schizophrenia. My career started in schizophrenia, and I still do work in schizophrenia. And we were interested in these neurophysiological signals, but Satish actually has one of the cleverest minds I know. He's now the president of King's in London, England. And Satish, one of his words to me was, Jeff, the motor cortex is of interest, of course, but it's really the dorsolateral prefrontal cortex that you need to go after, or the prefrontal cortex writ large that you need to go after for the treatment of depression. And so I took that very seriously, and I had an 18-month-old son at the time, and I flew to the only group in the world that was doing neurophysiology from the frontal cortex using TMS and EEG. So it was not a simple solution. The engineering behind using TMS and EEG combined is extremely complicated, because TMS stimulates and produces a magnetic field, which gets converted to an electrical field. And when you're recording EEG, you're recording these really delicate neurophysiological signals from the cortex. And so slamming the brain with a big magnetic pulse and then trying to retrieve these delicate neurophysiological signals is real complex. So I spent a month working with the folks in Finland, Risto Monami and colleagues in Finland, learned a lot, came back to Toronto, then had the good fortune of working with a colleague of mine, Paul Fitzgerald. We worked for several years together trying to work on these neurophysiological mechanisms, and happened to bring on some extremely talented trainees, including Itai Hadas, who worked with us. And what Itai did was he reversed engineered the neurophysiological signal from the cortex to be able to ascribe a signal to the subgenual cingulate, which you can see there in this slide. So what you see is that those red colors are physiological signal in the subgenual cingulate extracted from stimulating the dorsolateral prefrontal cortex.
And recorded in the scalp. Sounds like a big, hard thing to figure out.
It is very hard to figure out, and it wasn't us who figured it out. It was actually Silvio Casarato and the group in Italy that had figured out how to do this. And so we borrowed those methods and looked at depression and looked at our clinical outcomes in depression.
And how did they know the signal was coming from the reception?
Good question. So it was based on, you know, the what they call the inverse model of engineering, and the assumptions are complex, but I think they were able to favorably deduce that the two were related. In other words, that physiological signal from the subgenual. Now, your question is a good one though,
Because it's
because
a deep structure and
deep
it gets
structure.
volume conducted.
That's
widely.
correct. Yeah, it's complex. I will tell you that we've just recently ran, in fact, this paper is on my desk. We recently ran an analysis between functional neuroimaging and this TMS EEG neuroimaging, and there is a strong relationship between the two metrics. And you wouldn't necessarily expect that because the bold signal is not necessarily a physiological signal. And so, you know, it takes a little bit of wizardry to be able to string these two things together. But truth be told, this does look like a physiological signal from the subgenual cingulate. And what this graph shows is the top from left to right, those hot colors are actually sham. So stimulating with TMS for therapeutic purposes does not change that signal. But when you stimulate with TMS, if you go down to the orange color, this is active TMS following a course of treatment. You can see the suppression of that hot signal, the red signal, down to an orange signal, meaning that we're suppressing that. And the yellow is a group of healthy controls. So the top left is patients with treatment-resistant depression. Top right is patients who get sham stimulation. The signal does not change. Bottom right is the group that got active treatment, and bottom left is our healthy subjects.
Mind if I ask another question?
Please do.
So, you know, the dorsolateral prefrontal cortex, the next subcortical connection is the caudate.
Yeah.
Have people looked at signals at the caudate?
Good question. It's actually the caudate, and this is actually, you bring up a real good point. This is actually it's not a monosynaptic signal to subgenual cingulate, it's bisynaptic. And in fact, the first launching spot is caudate and hippocampus. So we have looked at the hippocampus. We're interested in that, and Itai was working on some really interesting data around hippocampus and knowing the physiological signal in the hippocampus because it's so pertinent to ECT. We haven't looked specifically at the caudate.
Okay.
No. This is more data, recent data from TMS studies. Top is Itai, I've already introduced him. Corey is at the bottom, and this person in the middle is Greg Applebaum. Greg joined us from Duke. Sometimes things just line up. When I first took my job at UCSD, they were trying to recruit Greg's wife, who is in the design center, and one of the senior academics at UCSD asked me if I was interested in working with Greg because they were interested in hiring the husband and wife, and Greg had neuromodulation experience and had already an incredible track record in TMS. And so we were lucky. We were lucky because his work aligned so closely with our work. And so the three of us have been working collaboratively. Itai has since moved on, but it's now Greg who's the director of research for our neuromodulation center, and Corey's the clinical director. And we work to generate this data, which is for all intents and purposes a replication of the original data, just in a separate data set. And you can see that really nice suppression of subgenual cingulate activity in response to treatment with TMS. Okay, pivoting a little bit to magnetic seizure therapy. So in 1999, I persuaded my boss at the time, who got me into research, to send me on a conference. And I went to a conference in Prague. The first conference, one of the first conferences that I ever attended was clinical neurophysiology, not a psychiatric conference. And at that conference, Holly Listenby and Harold Sackheim got up and presented the ability to produce a seizure. Now, that was about 2 or 3 months after I started doing ECT. So they were using already in 1999, they had developed a device in partnership with Thomas Schlapfer to be able to produce a seizure through magnetic fields. Why is that important? Because you can see there, you see the hot colors with electroconvulsive therapy. What happens is when an electrical field strikes the skull, it gets shunted. And then the CSF causes it to get volume conducted. And because of the resistance, you require lots of electricity to basically enter the brain. And it spreads throughout the brain. With right unilateral ECT, you certainly see that level of activity. But with MST, you see a fraction of the activity needed to produce a seizure in the cortex. This is another example. This is work by Holly's group, Holly Lisenby, who did some of the most seminal safety studies of magnetic seizure therapy. And you can see the not only at the surface level, which you see here, but these are E fields that spread throughout the brain and show you that when you're stimulating bifrontally and bitemporally with ECT, the brain is basically saturated with electrical field, and that electrical field spreads throughout and affects most notably the hippocampus, which we talked about earlier. But the hippocampus, of course, you all know, is associated with memory formation, and it disrupts the neuroplasticity of memory formation, and is probably the reason why patients get anterograde and retrograde amnesia, in addition to post-ictal confusion following a course of ECT. With magnetic seizure therapy, you simply don't see that. The field is very, very limited to the motor cortex. You can see there, and it's about 5 to 10 times less intense than ECT is.
But you get cognitive effects with.
Oh, well, wait for the punchline.
Okay.
So this is the development. This is the development of our timeline. As I mentioned, MST was developed, the first human trials in 2000s. It's been a real slow timeline. We typically say that, from initial discovery to clinical impact takes about 15 years. Well, we're already at 25 years, and we just submitted our paper comparing electroconvulsive therapy to ECT, and it took us a couple of years just to get it past the NIMH and to get it past FDA, and I'll tell you about that trial in a second. But this is the trial that led to that big trial. This was an open label trial of magnetic seizure therapy, over 100 patients, and we were looking specifically at different frequencies, because with TMS you can, not dissimilar to ECT, but with TMS we were specifically looking at frequencies, and what you see is remission rates in patients who receive magnetic seizure therapy. Again, open label, not randomized to placebo, and not randomized to electroconvulsive therapy, but favorable rates of remission in this population, specifically when they stayed on the protocol throughout the entirety of the treatment. I will highlight here that these were the way it worked in Toronto, and I think it's the right way of doing things, of course, is that patients first get TMS. We didn't have ketamine at the time, but patients first get TMS, and if they fail TMS, just because it's a lot simpler than and more preferable for a patient to get TMS versus magnetic seizure therapy, which is a lot more invasive, requires general anesthetic, et cetera, et cetera. And so these were, not only were they medication resistant, but they were medication and treatment resistant from TMS, which is why I think we see a somewhat muted therapeutic effect. So here's the answer to your question. Cognitive outcomes. So a laborious list. We really boiled the ocean on this one to look for cognitive effects. And what we saw was the BVMT recall actually improved. So not only did cognition not get worse, it actually improved. The AMI-SF, which is actually a poor tool to measure autobiographical memory, which we've abandoned. Now we use the autobiographical memory test. The AMI-SF did go down, but it did not go down significantly. And when you look at pharmacological trials looking at the AMI, and pharmacology isn't supposed to affect autobiographical memory, it deteriorates because the way AMI is pursued, it deteriorates over time. And so,
AMI is though a controversial issue, as you probably know.
Yeah.
You could see it in people with major depression,
right,
who've never had ACT.
that's right, exactly. So the AMI deteriorated, but the deterioration was not profound. Now, clinically, patients did not report any subjective cognitive impairment. They actually woke up faster than our ECT patients, which actually required us to modify the succinylcholine doses that we were using because they'd wake up and they'd be fully alert and awake real quick. And in some cases, they had wakening paralysis, which was quite frightening for them.
Yeah.
So we have significantly lessened our succinylcholine because of those observable effects. But all this to say that probably the only cognitive outcome here that is of relevance was a slight improvement in their memory recall.
Well, you know, with KCT though, there's actually an improvement in cognitive functions, memory lags behind them.
Memory lags behind.
Right.
Right. So this is suicidality. I'll touch on that in a second. So this is suicidality specifically, and suicidality also improved. But interestingly enough, it was the lower frequencies also used to induce seizures, but it was a low frequency specifically that produced the greatest rates of suicidality remission. So Holly and her colleagues published the first trial comparing ECT to MST, and these are the results. They show that there was near overlap between magnetic seizure therapy and electroconvulsive therapy. You can see the total responders and remitters not differing necessarily between two groups. So this was only about 80 or 90 patients. But it was the first attempt to look at the differences between ECT and MST. We did some clever neurophysiology, as I mentioned to you earlier. Yinming Sun, who's now actually with us at UCSD, is a neurophysiological whiz. He's now our neurophysiological expert. He was my graduate student in Toronto. And what Yinming was able to do was look at neurophysiological predictors of treatment response, and found that interneuron activity, inhibitory interneuron activity in the cortex predicted suicidality remission with MST by over 90%. In other words, you could use their neurophysiology to help predict who went on to achieve remission of their suicidality with MST, which is actually the data that we used we were able then to launch and convince the NIMH based on Holly's data, based on some preliminary data from our group, the open label data. We were able to launch this confirmatory efficacy and safety trial called CREST. It was a 5-year study that ended up being 8 years long because of COVID. Target was 260 patients. They were randomized to magnetic seizure therapy versus right unilateral ultrafrequency CT. There were 2 treatment outcomes of relevance. The first outcome was efficacy. We wanted to show non-inferiority between the 2 treatment outcomes. The second was cognitive superiority. So this was a gated analysis. We had to show, to convince the FDA, we had to show both evidence of non-inferiority between ECT and MST, but cognitive superiority between MST and ECT. In other words, MST being better cognitively. I don't have the data to show you. They're embargoed because we submitted them to a journal. But I will tell you, and the ultimate analysis involved 206 subjects. 206, we didn't quite get to 260. The only reason we didn't get to 260 was because of COVID. COVID slowed us down. We had to shut down in Toronto. It resulted in, we stopped doing ECT for about 6 months. But because we were slowed significantly, I had moved at the time to San Diego. San Diego's ECT service was real small, but it gave us, there's obviously not a lot of positives that came out of COVID, but my neck of the woods, it gave us the time to really enhance and develop the ECT service, and we were able to come on as a site. So Toronto was a site, UT Southwestern and Dallas was a site, and then finally San Diego came on as a site when we recruited the last cohort of patients. Because the trial went on so long, the NIH was getting impatient with us, rightfully so. Although if you look at our recruitment trajectories, we always met our recruitment trajectories until COVID hit. And so that's when things started to trail off. Be that as it may, we stopped the study at 209 subjects. And as I mentioned, those trials are currently submitted. I will tell you that it did reach non-inferiority, statistical criteria for non-inferiority. In other words, 2 treatments did not differ from one another. And I will tell you that it also reached cognitive superiority. MST not only did it not produce changes in cognition, but actually we replicated that original memory finding, that memory actually improved with magnetic seizure therapy. We did see a decline with ECT. So ECT did cause a decline in autobiographical memory, as well as other forms of cognition. Last bit of intriguing data that I'm hoping we will generate a study from is data that I worked collaboratively with Jenna Trainer. She was my postdoc in Toronto. She's a neuropsychologist who ended up now is in Boston. And we were real interested with MST because we had these suicidality effects with MST. And we thought to ourselves, well, MST does not impair cognition. In other words, it can be used, probably be used effectively in combination with psychotherapy. And because of the anti-suicidal effects, we were interested in this complex disorder of suicidality or suicidal behaviors in borderline personality disorder. We saw many cases of borderline personality disorder with severe depression and suicidality. Toronto responded really favorably to ECT. And because of our preliminary data, that enabled us to launch this trial of magnetic seizure therapy in severe depression with severe suicidality, borderline personality disorder. And what we found was a small trial. This trial was funded by the Brain and Behavior Research Foundation. What we found was a significant reduction in depressive symptoms, and we also found a significant reduction in suicidality in that small cohort of patients. But it was a profound effect that we're hoping to replicate. Jenna and I are collaborating on a trial of TMS for suicidality and borderline personality disorder that has been submitted to the NIH. And in fact, the results should be out soon. But I think we're eventually going to evolve those findings into a trial of magnetic seizure therapy. Hopefully MS3 will be approved in the next year or so. As I said, early in my career, I spent a lot of time looking at schizophrenia, and this was a pivotal trial by George Petridis, who conducted a trial randomizing people to clozapine and clozapine plus ECT. Clozapine is a fascinating compound that we had done lots of neurophysiology with and showed that clozapine really targeted certain receptors, inhibitory receptors in the brain. Clozapine, as you know, does not cause any signs of dopaminergic blockade. It does not cause any EPS, tardive dyskinesia. In fact, it's used favorably to treat tardive dyskinesia. It's a dibenzodiazepine pharmacologically, and it probably targets GABAergic receptors. We have published on that. But it produces a really unique physiological signal when you're recording EEG in patients with severe schizophrenia. And so the combination of clozapine plus ECT resulted in a very favorable therapeutic response in treatment-resistant schizophrenia. We had done a small study of magnetic seizure therapy in resistant schizophrenia. Victor Tang is another one of my fellows who is now faculty, was a fellow of mine, a clinician scientist trainee of mine, psychiatrist, and now faculty at the University of Toronto. Looking at the effects of magnetic seizure therapy in schizophrenia. And again, what we see is significant therapeutic improvement in psychotic symptoms in patients with schizophrenia with virtually no change in their cognition either. That led to a trial, a federally funded trial by the Canadian Institutes of Health Research with a colleague of mine, George Lucius, and he's a professor at the University of Toronto. Unfortunately, once that trial got funded, I had to step away and move to San Diego. I didn't have to, I wanted to. But that trial is ongoing right now at the University of Toronto. I will finish so I can leave time for questions. There's my summary. I think we have lots of neuromodulation tools right now that are working very favorably in resistant depression. I think resistant depression is a lot more common than we think. We see it all the time in clinic. Pharmacological strategies are effective, but they often fall short. And not only are we getting really making breakthroughs in treatment-resistant psychiatric symptoms, but I think we're not that far away in terms of biological signatures of treatment response. And we're currently conducting, as are many others, numerous multi-center clinical trials trying to ask those questions. This is our program. Corey Weissman, who you know through these slides. JP Miron, also a fellow in Toronto who we recruited to UCSD. Those are the two folks who are leading our clinical research services. Beth Twomley and Jyoti Mishra are scientists working closely with us. We currently have TMS in Guava, which is our research center. Dove Canyon is our outpatient center. It's the hub of our neuromodulation service. Down at Hillcrest Hospital in La Jolla, and we're about to start. We started ECT at the Alvarado Hospital, which is now called East Campus. And I think will be one day our hub for our neuromodulation service, because it's going to be our hub for mental health. Lots of studies that we're currently conducting, both clinically, but also in relation to clinical trials. Those are our timelines. Lots of naturalistic trials, and we have different trials currently underway across the country. And these are just some of our trials: CREST, TMS for hoarding, PAC TMS, which involves treating depression and cognitive training in patients with TBI. FREED was our trial looking at individualizing TMS towards neurophysiological signatures. Corey has a K for accelerated bilateral TMS for suicidality. We finished our trial in bipolar disorder, and that was published. I didn't present the results here. In our Rockaway trial, I did present some of the results here, looking at accelerated TMS for depression. And you know, what can I say? That's La Jolla. It's one of the most beautiful places on earth. And thank you for your time today. For this. Wow.
All right, I'll scoot back in here so people can see me. So thank you very much for that. Folks at home, yeah, so we started a little bit late, so we'll stay on here a little while for questions. If any of you watching would like to ask anything, go ahead and type it in to that Q&A there. Yes, you can. We don't have any online yet, so why don't we open it up to the room? We have about 7 or 8 people here in the room, so yeah, go ahead.
Yeah, so this is an ongoing debate about, is it the seizure with ECT or is it the stimulation? What's your take on it?
I think it's the seizure.
Okay.
And the reason I think it's the seizure is because with magnetic seizure therapy, you can produce a seizure with 400 pulses, 100 hertz for 4 seconds at real high intensity. It's really unusual to produce significant neurophysiological effects with only 400 pulses. But once the seizure hits, then the seizure basically takes over and and the seizure itself is producing LTP in the brain. Those neurons are firing together. You can see that on the EEG. In fact, millions of neurons are firing synchronously, which is why you get that unique and distinct spike wave activity on EEG. I really do believe it's a seizure. I don't think it's just, you know, stimulating the brain for prolonged periods. I think that's how it works with TMS, but I don't think that's how it works with electroconvulsive therapy. I think there's something unique in the seizure. And in fact, the seizure itself produces this spike wave complex. And I presented this in other, I've showed this data in other presentations, but I have not shown it here. It's that spike and the subsequent wave, which is a GABAergic wave, and that GABAergic wave builds over time, and it's what ultimately terminates the seizure. We have a trial right now that is being reviewed where we think that with TMS we can actually reproduce that neurophysiological wave, and if we personalize the number of TMS pulses that we deliver, we can enhance that inhibitory plasticity, and we think that's the key to treating depression. So that's a trial that it just got funded by BBRF, but we're hoping that in the next year it'll be funded by the NIH. And the goal will be, rather than seeing TMS as 600 pulses, it's actually a continuum and it's individualized per person. So everybody will have different frequencies and different stimulus numbers to be able to achieve a more robust therapeutic response. So to answer your question, it's both. But in ECT, it's definitely this. I think it's definitely the seizure. With TMS, I think it's pulses. Questions here, Drew?
Can you comment on the right-sided versus left-sided dichotomy of frequency that's been reported? And I've seen some studies that are non-inferiority designs that everything works. And do you believe that the right versus left dichotomy is what's that based on?
Yeah, I think that it is the right side can produce therapeutic effects in and of itself. So if you stimulate over the right dorsolateral prefrontal cortex, you can produce therapeutic improvement. We've known for years that when you stimulate over the right side with ECT, stimulate on the right side produces therapeutic effects. But stimulating bilaterally produces more robust therapeutic effects. And so I'm actually, if you look at the meta-analyses in TMS, time and time again, the meta-analyses show that when you stimulate bilaterally with TMS, you get more robust therapeutic outcomes. So I think there is something to the right side, and specifically in our data, the left side had very little to do with suicidality. It was when you introduced bilateral TMS that you produced not only the greatest depressive symptoms response, but you also produce remission of suicidality.
I guess I meant the fast versus slow on the right. Do you think that there's any traction to this, to the frequency of 1 versus 10 hertz?
Great question. No. I don't think there's a shred of evidence that stimulating with low frequencies over the right side in the spirit of inducing suppression is bona fide. In fact, in the 90s, when they originally came out with this idea of this dichotomy between high left, slow right, it was based on this, I think, probably quixotic notion that the right side needs to be suppressed and the left side needs to be suppressed and the right side needs to be excited. Sorry, the other way around. All this to say that when we looked at those meta-analyses and of fMRI, there wasn't a shred of evidence that suggests that depression is hyperactive over the right side and hypoactive over the left side alone. There is hypoactivity, but it's bilaterally throughout the brain. So, Alvaro Pascual-Leon is one of the leaders in the field. Alvaro published some studies in the early days showing that if you stimulate with high frequencies over the left side, you can potentially produce a mood dampening effect. And that's why everyone avoided it. We had conducted numerous trials with bilateral hyperactivity in patients with depression. Their moods were just fine. So I actually think that bilateral high frequency stimulation is something that is unanswered and needs to be carefully considered.
Well, let's go to our one question from someone online asking what's keeping us from doing MST.
Regulators. It's not approved yet by the FDA. My hope is that it will be approved. So I was speaking to Jürgen yesterday, and when we got this trial funded, this CREST trial funded, maybe I'll talk about it at our lunch because it was an interesting path. We originally were interested in suicidality because, as you know, the NIMH basically lost interest in clinical trials for treatment of depression. And so we had to use these this is 2015, we had to use these RDoC targets. And so they were willing to consider things in suicidality. And so we submitted this big trial in suicidality. And then they said, okay, you've been funded. Now we want a trial in depression. So we rewrote the entire thing to treat depression. Then we registered it with the FDA. That whole process took us a year. And we had to convince FDA reviewers who were a lot more critical and scrutinizing compared to the NIMH reviewers. But it took us a year to get that trial funded. And we had to get funded by the NIMH, we had to receive approval by the FDA that this was registered as a clinical trial through the FDA. In other words, once the trial outcome was identified, those data were the approach that we took satisfied FDA reviewers, and now shortly they'll be reviewing the outcome of that trial to determine whether or not MST should be approved by the FDA. And then, as you know, CMS will look at that data, and then insurers will look at the data. So we're still a bit of a ways off, but I'm optimistic that we'll have an answer next year. So. Great.
All right. Well, thank you for being here. We
Thank
can
you.
continue this discussion at lunch. And folks online, thanks for joining us. I didn't mention it at the beginning, but we are recording today's presentation. It will be available on our archive if you'd like to revisit this at any time. So until then, thanks for being here and hope to see you again next week.